Hi everyone. So for those who are here, you can scan the QR code for your attendance. Okay, thank you. Mohon untuk buka kamera Thank you. Good afternoon, Pavitran. Semua dah makan? You had your lunch? Alright. Shifa kenapa tak makan lagi? Tak apa, 1 hour only. Okay, and then you can go to eat. Izzah pun belum makan. Tak apa, kejap je. 1 hour, 50 minutes je. Okay? Tin Hee, good afternoon, Tin Hee. Okay, 1 more minute and then we will start our lecture. Yeah? Okay, I will put the QR code later on. Yeah, so I'm starting to share my slides now. Kejap. Where is it? Okay. Okay. So, can anybody give me a confirmation whether you guys can see my screen? Can I get a confirmation? Okay, Angie said can, doctor. Okay, thank you for your thumbs up. All right. Okay, so now we are going to be moving on onto the new topic in our course. All right, so the topic is the forensic serology and DNA analysis part 2. All right, so previously we have learned about part 1 on the presumptive test, and also we have looked into different kind of confirmatory tests and what do you do when you want to collect the samples, or in this case the evidence. All right, okay, so let me okay, all right, so now we are going to have a little bit of recap for our first lecture on forensic serology. Okay, anybody want to try? Okay, any volunteer? We just recap on whatever that we have learned before. I think on Monday. Yeah. All right. Anyone want to try? Michelle, do you want to try?
uh, we talk about the DNA, how to like knowing the blood is human blood or animal blood, and like define is it blood or actually it's just a pigment, a red pigment or something like that.
Okay. All right. Good. So basically, now we are going to have a little bit of recap. So previously for forensic serology part 1, we have looked at into what is the constituents of blood. Okay. What are the constituents of blood and how do we determine whether when we arrive at the crime scene, whether it is a blood or it's just a red liquid, for example, in the case of blood. Okay. So that's how we do the luminol test for blood. Okay. And then we also looked into on how are we going to differentiate between whether it is animal blood or just human blood. That's the second thing, which is the precipitin test. All right. So based on this picture, okay. So let me get my laser pointer. Okay. So this picture depicts on how are we going to synthesize the human serum to be used for apa? For the precipitin test. All right. Okay, so after that, we have moved forward onto the use of other biological fluids as well. Other biological fluids such as semen, seminal fluid. All right. So for this kind of biological fluids, also we do have the presumptive test, the confirmatory test as well. All right. So because we don't want to bring back to the laboratory of something that is not even the evidence that we are interested in. Okay. All right. So basically that is forensic serology 1. So now the continuity of it. So now you have all of these biological compounds. You have your blood, you have your semen, you have seminal evidence. So now we are going to further it to do DNA analysis for identification purposes. Okay, so now we are going to look. I'm sure that you already know that. After this, we are going to look into what is DNA is all about and how do we do the PCR, polymerase chain reaction, how do you amplify it. Okay, so after this, we will look it into detail. All right. Okay. All right. Okay. So at the end of this topic, you should be able to differentiate between what is RNA, what is DNA, what is nuclear, and what is mitochondrial DNA. What are the differences between them? Okay. And then you would also need to be able to explain the basic principle of polymerase chain reaction, which is which is very important process in amplifying your DNA for forensic purposes. Okay, so I cannot stress it enough. PCR is very important. So you need to be able to understand the basic principle and you be able to explain the basic principle of PCR. Okay, all right. So moving on. All right. Okay. So what is DNA actually? DNA. DNA stands for deoxyribonucleic acid. You have now apa during this pandemic you heard about DNA, RNA. So what are those things? Okay, so DNA, I believe that you have learned this a little bit during your biology classes. Okay, so DNA stands for deoxyribonucleic acid. Okay, DNA. All right, so how about RNA? RNA is just ribonucleic acid. Okay, so that is the differences between the first differences, which is the name lah. Okay, but we will look into depth on the structure and everything. Okay, all right. So DNA, deoxyribonucleic acid. This is, as you see at my pointer here, you can see this is the basic structure of DNA. Okay, the basic structure of DNA comprised of deoxyribose sugar, which is the center of the DNA, and then it is connected to a phosphate back group. Okay, phosphate group, and then it also connected to a base. Okay, so base here is very important lah. Okay, so this phosphate and deoxyribose sugar will act as your as the DNA backbone. Okay, base ni we will look at into later on. Okay, so all right. Okay, so the base we will look at after the four types of different nitrogenous bases for DNA. All right. So when we are talking about DNA, what does DNA do? Okay, DNA contains genetic information. Okay, you would imagine yourself inside the cell. Okay, you go inside the nucleus. You go inside the nucleus, and then inside the nucleus, there are a lot of DNAs inside your nucleus. So DNA contains genetic information of yourself. Okay, so this genetic information, it codes for protein production in your body, which is what maintaining your livelihood lah. Alright, so DNA contains genetic information for the production of proteins inside our bodies. Okay, that's the first thing that you need to know. What does DNA do? That is the basic functions of DNA. Okay, so this is the structure of the actual DNA. We have looked into the DNA punya bases here. Why is this not going? Okay, so we have looked into the basis of DNA. So now we are going to look at into this is the actual shape of DNA, which is in a helical form. Okay, helical form is something the DNA will exist in a double-stranded helical form here. You can see that from this one backbone, which is phosphate and sugar, is going this way. Okay, and the second strand is going this way. One is going to the left, one is going to the right. And both of these strands are connected by a base. So these bases, these nitrogenous bases, is the adenine, thymine, guanine, and cytosine. Okay? So all of these four bases have their base pair rule lah. We look at it at A, adenosine is going to always be paired with thymine. Okay, so you can see here, A is always going to be paired with thymine. Thymine, of course, A, vice versa. Guanine is also always going to be paired with cytosine and vice versa. Cytosine with guanine. Alright, so these bases, when they are connected to each other by hydrogen bond, it creates this helical shape. Okay, alright. Okay, so now moving on to the next slide. Okay, so as I mentioned before that DNA encodes for our protein production. So how does DNA encodes for our protein production? Macam mana DNA from inside the nucleus form protein? So we are going to look into it. Okay, so DNA, as you see that, okay, is it helical form, okay? And then DNA would undergo transcription. So what is transcription? Transcription is a process where RNA is made up from DNA. So now we are creating RNA from DNA by transcription process. So this process of transcription happens inside the nucleus because the housing of your DNA is inside nucleus. Okay? After transcription, the RNA will be continue going. So RNA ni daripada inside the nucleus, it will go out from the nucleus and go into the cytoplasm to the ribosome. Okay, near the ribosome, RNA will will undergo translation process. So translation process is where your RNA ni is being encoded onto the protein. So RNA, whatever message that has been carried by this RNA, it will be processed by the ribosome under the translation process, and this will create protein. So that's how DNA goes to RNA, goes to protein. Okay, by transcription and translation process. All right. So, okay, among humans, most of the 3 billion bases that researchers have been mapping out, the bases of DNA for humans are exactly the same. Okay, basically, what are the numbers? Now we are looking at the numbers. All right, our human DNA, approximately 99.7% is similar to each other. So for me and you, our 99.7% of our DNA is the same actually. But only 0.3% from us is different. So this we use this 0.3% to differentiate from one another. Okay, that's how our body works. Okay, that is what has got given to us lah. All right. Okay. So no two people would have identical DNA. This statement might be correct and also can be wrong. Okay. So no two people have identical DNA is wrong. You need to be followed by except identical twins. So no two people have identical DNA, yes, except for identical twins. Identical twins would have identical DNA because of their formation itself, the separation of the zygote, creating two embryo inside your mother uterus. All right? Okay. And there is also another condition that we call okay. No one people have two different set of DNA. Okay, can you imagine that people have two sets of DNA? Okay, so there is no two people have two sets of DNA. True, false, except for chimera and bone marrow recipient. Okay, so what does this mean? Okay, now we are going to look at into how chimeras and bone marrow recipient differ from each other. Okay, so basically chimeras and bone marrow recipient ni there are medical conditions lah. Alright, so basically for chimeras, it is caused by a twin dying early in utero. So basically, they have two sets of DNA inside them. Okay, maybe the sex cells have another DNA, and other somatic cells also have another DNA. So basically, two sets of DNA due to this condition. All right, this also happens to bone marrow recipient. Okay, bone marrow recipient, you know that bone marrow recipient transplant. So bone marrow is the housing for the production of your blood. Okay, so this housing of your blood would have a different DNA as compared to the rest of your cells body. So in this case, there are two. There is no one person that have two set of DNA except for chimeras and the bone marrow recipient. Okay, so that's why the statement is very important. No two person have identical DNA is wrong, except when you add except for except for identical twins, then it's correct. Okay, all right. So now we have learned that there are some cases in the world that have two set of DNA and have identical DNA, but the rest of us would not have identical DNA and would not have double DNA inside our body. All right. So okay, so this is what I've explained before. All right. So let's see what Michelle said. Michelle, isn't it? Oh, I think I cannot see the chat now. Okay, maybe later on, I will look into it. All right, thank you, Michelle. All right, so basically, this is the composition of DNA that I've talked about. Okay, sugar phosphate backbones, and we have nucleotides. We have the base pair. All right. So this is the structure, the double helical structure. And then we also have looked into the base pair rule. So adenine always goes with thymine. Okay, you just need to remember ATGC. All right. A always T, T always A, G always C, C always G. Okay, so you would not have A, C, or T, G. All right. So this bases, okay, for example, A and T here. So this basis is connected by hydrogen bonds. Okay. All right. Okay. So, okay, this is our first class activity. All right. So, on the one side, we do have A, T, A, T, C, A, T, G, C, G, G, G. All right. Who wants to try on what is the other side? Okay. Who wants to try? Okay. Volunteer. Sotling, do you want to try?
Can doctor?
All right. Give it a shot.
TAT,
Okay.
AGT,
Good.
ACG,
Good.
CCC.
All right, good, good. So it's go. So basically, it is TAT, AGT, ACG, CCC. All right. That's good. Okay. So basically, now you have not confused with the base pair rule. All right. Good, good. Thank you, Solling. All right. So now we have talked a lot about what is DNA, what does it do. So where can we find this DNA? Previously, I have said to you that DNA exists in a nucleus of the cell. All right. So all types of our cells contain a copy of the same DNA. So would you imagine that all of the cell inside our body have the same genetic information? Our skin cell, our hair cells, our all our tissues, our organs, all of the cells have the same composition of DNA. Okay, therefore, based on this, we can deduce that what are the cells that are important in forensic science. Okay, so inside forensic science, white blood cell is very important, sperm cell is important, and chick cell is very important because all of these cells contain genetic information. Only red blood cell does not have any nucleus, which means that it does not have any DNA. Right, that's the thing that you need to remember. So when people ask what kind of cell do have DNA, white blood cell, sperm cell, chick cell, but not red blood cell. Okay, red blood cell, the function is only to carry oxygen. All right. So, what are the sources of DNA at crime scene? So now you need to know where can you find this DNA at the crime scene. So previously you have looked into when we have looked into the forensic serology one, we have learned that okay, you can have blood, semen, saliva, tissue, bone. Bone is something new. Okay, bone, teeth, hair, maggot crops. So all of these substances, all of this evidence contain DNA for you to be able to take it much more further for DNA analysis. All right. All right. Types of DNA. Okay, I might go to the next slide first. All right. Types of DNA. There are two types of DNA, which is nuclear DNA, which is, as it names, it is inside the nucleus. There is also a mitochondrial DNA, which can be found inside the mitochondria lah. Okay, so mitochondria, where does it located? It is inside the cytoplasm. Okay, so there's something interesting for mitochondrial DNA because mitochondrial DNA is inherited only from the mother. Okay, and this is also important that mitochondrial DNA is very rich in skeletal form, inside the skeletal cells lah. So that's why, for example, if you have found skeleton at a crime scene and you don't have anything else in terms of biological evidence, then you can use those skeletal to get your DNA. So because skeletal cells have abundance of mitochondria and therefore you have abundance of mitochondrial DNA. Okay, so how can we say that mitochondrial DNA is inherited only from the mother? Macam mana kita boleh kata it is only from our mother? Why? Our father don't have mitochondria? Okay, because it goes back to to the fertilization process. Okay, all right. So now you have looked into this. Okay, this is the egg cell from your mother. So you have a nuclear DNA, and of course, inside the cell, they must have a mitochondria. This is the sperm from your father. All right. So you have a nuclear DNA at the head of the sperm, and the mitochondria is at the neck of the sperm. All right. During fertilization process, the nuclear DNA from the father will combine with the nuclear DNA of your mother. All right. Leaving the father's mitochondria outside the cell. So only the genetic information from your father would include inside and fertilize the nuclear DNA of your mother, but not the mitochondria. The mitochondria would of your father would just remain outside of the egg cells. Okay, so that's why only mitochondria from your mother exists in your body. So basically, we have a long lineage of um, maternal mitochondria in ourselves. So we don't inherit mitochondria DNA from our father. All right. So this is the reason. Okay. Okay. So, yes, so nuclear DNA yang tadi tu yang from fertilization process, it is inherited from our ancestors. But mitochondrial DNA is inherited from a single lineage of our mother only. Okay? All right. Okay. So, mitochondrial DNA, what are the advantages of mitochondrial DNA? Okay, mitochondrial DNA, the advantages is that it is much more sensitive as compared to nuclear DNA. Much more sensitive in terms that it does not need a much mitochondrial DNA to be tested. Okay, it can maybe just a small amount of mitochondrial DNA can be used for DNA analysis. All right, mitochondrial DNA also degrades much more slower as compared to your nuclear DNA. All right, so mitochondrial DNA can be used for apa? In where there is no nuclear DNA available. Okay, so as I mentioned before, if you have looked into the skeletal remains, if you have only skeletal remains but then no biological evidence to be found anywhere, so you can use the skeletal cells to get the mitochondria DNA. Okay, all right. Okay, so what are the disadvantages pula? The disadvantages is that all of the people from the same maternal line, okay, from the same maternal line will be indistinguishable because all of us have the same mitochondrial DNA. Okay, all right. So it is more work, more time consuming, and usually much more costly. Okay. So now we have looked into the DNA. We have looked into what are the types of DNA, where to find the DNA. All right. So I'm sure that you still remember that only 99.7% of our DNA is similar to each other, and only 0.3%. This 0.3% is something that makes us different from one another. Okay. All right. Okay, so now we are going to look into depth on the DNA itself. Okay, so now we are going to do the DNA analysis. Okay, so this is very important. Okay, so uh, as you look at the slides before, the 3 million bases that we said that available inside our DNA, 95% of those DNA does not code for any protein. Okay? So 95% of your DNA tu does not do anything. Only 5% creates protein. So only 5% undergoes transcription translation. Kan tadi yang initial the thing that we look to production of protein kan. On that only 5%. So 95% ni buat apa? So this 95% is something that can be used in terms of for your identification. Okay, so your 95% have different composition from my 95%. Okay, so okay, So how do we look at this? Okay, what makes me say that your 95% is different from my 95%, which leads to individuality. Okay? All right. Okay, now, okay, we are looking at the apa? At the DNA. Okay, we are looking at the DNA. So there are coding regions, and inside the DNA there are coding regions and non-coding regions. Okay, coding regions we call it exon. Non-coding regions we call it intron. Okay, so these exons, which corresponds for your protein production lah. So this intron, which is the non-coding regions, is something that you can use for identification purposes. Okay, this intron, we can also call it as a short tandem repeat. We will look it after this. Okay, so there is a lot of word jargon that you need to learn, that you need to know. Okay, exon lah, intron, and then now short tandem repeats. But this is important because for you to be able to understand on what is PCR is all about. Okay, so exon, okay, you just need to remember exon, okay, dah buang, tak payah fikir, because exon would not be used by apa, for your identification purposes, because it is for your protein production. So intron now for the non-coding regions. Okay, so from the non-coding regions, which is your intron, it helps in individual identification. That's why I said your 95% of junk DNA that doesn't do anything is different from my 95%. So how does this goes? Okay, so you see this first pakcik here, okay, this first man, okay. So you see that, okay, now we are using the same the same base lah, AGAT, okay, the same base. So for this pakcik, it has 7 repeats of AGAT, okay, AGAT, 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 and then you have your exon here, which corresponds to your protein production, and it continues AGAT again for 6 repeats. So this pakcik, this first man here, repeats AGAT for 7 times and then have some length here, that is your exon, and then repeat AGAT again for 6 repeats. But for this guy here, this blonde guy, it repeats AGAT for only 4 times. And then you have a long exon. And then it repeats AGAT again for 19 repeats. As what this brunette guy here, okay, the red hat guy here, it repeats AGAT for 12 repeats, and then you have a small exon here, and then it repeats AGAT again for 14 repeats. So all of this person have different composition of intron and exon. So these differences in intron and exon would create your identification. Okay, so all of this, we call it this short tandem repeat. Okay, this is short tandem repeat for 7 repeats. This is a short tandem repeat for 19 repeats. Okay, all right. So all of these short tandem repeats will be used for your DNA analysis in your PCR. Okay. So what are the basic steps in DNA analysis? Of course, we do have extraction. Okay, so we separate DNA from our sample, and then we do amplify it. So this is the one that I've said, amplify using a PCR, polymerase chain reaction. And then we do have separation, profiling, and fingerprinting after that. All right. So in terms of extraction, you have looked into the DNA extraction into 3 different stages. Okay, so basically we extract cells, so the cell will be ruptured or undergo lysis, and then this cell would undergo protein denaturation, because inside the cell there is a lot of substance that protect the nucleus. And then lastly, after we have do the protein denaturation and inactivation, and then we have the extraction of DNA. Alright, so now what do we do with this DNA? Because DNA yang sekarang ni is just that one DNA from one cell. Okay, so we are going to do polymerase chain reaction, which is an amplification. We amplify our DNA to a number that is easier for us to being detected. So you have also heard about PCR during this COVID-19 pandemic kan. So this is what they do. Okay, so basically your double-stranded DNA will undergo three stages, which is denaturing stages, and then it undergoes annealing stages and extending stages. All right. So I think that this is much more better visualized using a video lah. Okay, so I will try to. Okay, all right. Okay, first of all, can you see the video? Boleh nampak tak video? And then can you hear the video? Tinjing, boleh dengar ya video ya? Boleh, alright. So I'm just going to play ya. Okay, let me just pause a little bit. So now you from this video, you know that in the basic ingredients for PCR is that okay, you need to have your target DNA, which is your short tandem repeat tadi lah. Okay, short tandem repeat your target DNA, and then you would need to have primer as well. And you also need to have your nucleotides, the bases, the nitrogenous bases that we have talked about, ATCG. So these bases would need to be available for amplification. So how does this happens? By DNA polymerase. Okay, DNA polymerase ni is an enzyme that is being using inside the PCR reaction. Okay. Okay, so basically the primers is something that you as your starting point. Okay, so you would have to decide on what kind of primers to corresponds to the STR bases that you have looked about. Okay, so these primers will act as a starting point here. We see that this is the starting point for 3 prime to 5 prime, and 3 prime to 5 prime here. This is the starting point. So the DNA polymerase would just take up the bases, available bases inside the solution, and just put it down back together to create some new strands. Okay. Okay. So basically, PCR, that's what I've said before. Sekejap, okay. Can you see my slide now? Nampak tak slide saya? Anyone can respond? All right, thank you, Nuramira. All right, so basically, that's how PCR works. Okay, so from one DNA, it will be able to amplify after 25 cycles. It will create some sort of like a 25 million of the same DNA. So this amplification would help in the identification purposes. Okay, so all right. So based on the videos that you have watched, so now you have understand on how does the PCR is all about, what are the processes. Okay, based on the videos, I would need you to go to the Jamboard. I will give you the link now. Let me end my show first. All right, so go to this Jamboard and then suggest apa a case or application to be used for PCR. Sekejap, stop sharing. Okay. Alright. So, can you access to the Jamboard? Let me try first. Boleh tak access? Anyone can respond? Boleh ya? Okay, thank you Michelle. Alright. So, inside your inside the Jamboard, okay, please suggest a case or application of PCR. Okay, so now you have looked at okay PCR, what is PCR is all about, okay, and then whether PCR can be used on any other application besides beside forensic application pun it's okay. It's just that for you to be able to know what is the application of this PCR is all about. Okay, so you can use stickers and then as usual at the end of the your idea you just put your name. All right, you have 5 minutes. Okay, siapa ni? COVID test. You can put your name at the end of your idea. Okay. All right. Where are the others? All right, human identification by PCR, amplification, short ternary repeat on genome DNA. Okay, good, Melanie, good. Detect inherited disease, yes. So usually inside your inside your DNA, sometimes there are mutations, kan? So these mutations also can be used to detect for diseases right, such as cancer ke, or any kind of other diseases that is related to your genetic. All right. Okay. Any other? The modification of DNA fragments. Yes. So usually that is for the mutation lah. Okay. Mana lagi? Mana lagi? For cloning genes. Yes. So we can also do gene cloning. Okay. Polymerase chain reaction to diagnose. Food pathogen detection. Yes. Good. So it also can be used not for just humans, but also can be used for pathogens as well, for bacteria. All right, drug discovery and synthesis. Yes. So this drug discovery also can be used for PCR to tally with your genetic sequence. Okay. All right. Okay, that's good. Okay, so approximate we do have 2, 4, 6, 8, 10, 12, 13, 13 suggestions. All right, that's good. Okay, thank you for your suggestion. Okay, let's go back to our slides a little bit more. Okay. Okay. So basically, this is what we have looked before lah, on the polymerase chain reaction. So this is in terms of the words. So, but if you understand the videos, then these words will come naturally to you lah. Alright, so basically polymerase reaction is a technique to make thousands copies of your DNA. Alright, so million of copies can be made from a single speck of blood. So based on the video, as I mentioned before, one strand of DNA would be during PCR we will be able to create about 25 to 30 million copies of DNA after 25 cycles. Alright. Alright, each cycle takes about 2 minutes from start to finish. Alright, so usually for 25 cycles, about 1 hour lah. Okay, alright. Okay. So basically, this is also in terms of the diagram lah. So one template DNA, after the first cycle, you have 2 DNAs. After 4th cycle, and you imagine after 25 cycles, you have thousands of millions even of your DNA copies. Alright. Okay, so now we are going to do a little bit of Kahoot. Is it okay? You guys boleh lagi ke? Kita buat Kahoot sikit ya. Okay, sekejap. Alright, thank you Fatiha. Michelle said, okay, doctor. Good, good, good. Mana Kahoot? Alright. Sekejap ya, saya stop sharing kejap. Okay, we have 8 more minutes. Okay, maybe we can do a little bit more Kahoot. Slow pula lain sekejap. Okay. Loading game pin. Okay, now. You see the game pin? Okay. Okay, I think we can start now. Okay, it's still increasing. Okay, start ya. Okay, forensic serology. Okay, DNA contains genetic materials that codes for protein production. Is it true or false? Betul ke? Is it correct? Ke DNA contain for something else? This is an easy question. I'm just softening up to you. Alright, true. Yang salah, yang say false ni kenapa? 3 orang ni mungkin tersalah tekan. Okay, alright. Okay, so who's the first one now? Hang. Alright, tinging followed by tinging. Okay, next one. Which of the following is not the characteristic of DNA? Yang mana bukan characteristic of DNA? Which one? Saya dah sebut banyak kali. Which one? Yes, it is not a single-stranded. Okay? Alright, good. Okay, Jihan naik atas. Good, good, good. Okay, which of the following is not the base for DNA? Yang mana bukan base DNA tadi yang kita the thing that we have discussed for base pairing rule? Which one? Yes, uracil. And the thing that is interesting here, uracil is the base for your RNA. I have not discussed the bases for RNA kan. So uracil is one of the bases for RNA. So it's up to you now to find out which corresponds to RNA. Which uracil corresponds to apa benda. Okay? So that's your task. Alright. Okay, Pavitran menaik Melanie. Okay, no two people have identical DNA. No two people have identical DNA. Betul ke? Line putus. Alright, tak apa, tak apa. Okay, sekarang can everyone see the screen? Boleh nampak screen ke sekarang? Okay, Michelle said okay. Oh, never mind, never mind. This is just for your practice only, okay? Alright. So true or false? No one people have two different set of DNA. True or false? So as I said kan, this is wrong except kalau I put identical twins. Alright. So, Mir naik sekarang. Toby apa? Toby Maguire ke yang naik ni? Alright, which of the following samples cannot be used to source DNA? Alright, red blood cell, as I mentioned, red blood cell does not have any nucleus. Okay, nuclear DNA can be found in the cytoplasm and mitochondrial DNA can be found in the nucleus. True or false? You need to read it carefully. Alright, mungkin tersalah baca. Nuclear DNA can be found in the nucleus while mitochondrial DNA can be found in the cytoplasm. Okay, so Toby Maguire. Okay, mitochondrial DNA is inherited from our mothers. Is it true? True ke? I thought that it was from our father. Yes, okay, true. From our mother. Right, Toby is still at the top. Okay, there are what regions in our DNA? Tadi. The new word jargon that you have learned. Okay? Which one? Yes, intron and axon. Correct. Okay, Toby is still leading. Good, good, good. Alright. After 25 cycle of PCR, 1 million copies of the original DNA have been copied. 1 million ke, 20 million ke. So millions lah. We are talking about millions of DNA have been copied. Okay? True. Okay, our last question. Okay? Last question. DNA have repeating units called? What are the repeating units of DNA? For identification purposes. Aduh, hai, connection loss pula.